My name is Elvis, a postdoc at the Division of Human Genetics in the Faculty of Health Sciences at the University of Cape Town here in South Africa. So I would be the moderator for today's webinar for the African Pharmacogenomics Network. And we have the privilege to have Dr. Othman speaking to us on pharmacogenomics, beyond genomics, challenges and prospects from bioinformatics and artificial intelligence. Brief on Dr. Othman, our speaker, our guest for this webinar. He is a postdoctoral fellow at the Sydney Brenner Institute for Molecular Bioscience at Wits University in Johannesburg, South Africa. He has been involved in several projects that apply biomolecular simulation, molecular modeling, dogging to understand structure-activity relationship of anti-cancer peptides. His current research focuses on implementing structural bioinformatics methods within high-throughput genome annotation to predict the impact of mutations. His group has been contributing to this field by using this approach to characterize pharmacogenomic landscape of African populations and to predict the effect of genetic variability on drug response to improve and contribute to clinical guidance for drug indication in precision medicine. He also has interests in areas including data science, database development, and he is a promoter for open access practice in research. And he is also key in supporting bioinformatics education in low and middle income countries. So we'll have our speaker speak to us on the topic as indicated earlier, and I will entreat that we all keep our mic mute and also our videos off and use the Q&A tab to put in our questions. Depending on the time, we'll be able to have one or two inputs, which we will indicate by raising up your hand after we've exhausted the questions in the Q&A section. So Dr. Othman, we are privileged to have you. You have the floor if you are ready. Thank you very much.
Thank you so much. I'm very happy to be here today. So I know this is Saturday, it's a holiday. So if you are here, you're really motivated to attend this talk. So for that, I thank you. And because I appreciate that, I hope that I will not annoy you with a lot of details. And this is why actually I have made this talk in a way to be concentrated on the main idea. So rather than throwing some results from my research, which may interest you and may not also interest you, I have come with a central idea that we want to focus on. And I hope through the different slides that I will show that I will convince you with this idea. And it's actually something that is bothering me since, I would say, 2 to 3 years now. And I hope that I can highlight this issue. So first, I want to make sure that you are seeing my cursor, my red cursor.
Yes, we can see it.
Okay, perfect. So just for if you are impatient, that this is the what's the talk about actually. So what we are going to cover is I'm going to highlight a major challenge in pharmacogenomics implementation in Africa through some studies that we did before on genomics, and also that integrates a lot of different techniques, integrative analysis to be precise, and how could bioinformatics and artificial intelligence addresses these challenges. So this talk is actually composed of 5 parts. The first one is the prelude. Then I'm going to show what is the concept of complexity in pharmacogenomics. I'm going to then state the problem and then I'm going to show the input from bioinformatics and artificial intelligence. And then we are going to conclude and highlight maybe some solutions. So first, let me just give like the ground zero of pharmacogenomics, actually. What is actually a pharmacogenomics? So basically, what we are interested in is characterizing the genetic variability for some particular genes. These called ADME genes, actually. So ADME genes. And these ADME genes, they are subdivided into 4 main classes. So we have phase 1, we have phase 2, we have phase 2 enzymes, and we have transporters, and we have also modifiers. So for the phase 1, these are particular enzymes that will contribute in the breakage of some of the covalent bonds of xenobiotics and also drugs. And then the phase 2 drugs, they are basically, they do a conjugation of some polar groups to the ligands of the drug in order to make them more polar so that they could be extracted very easily in the urine or any other fluid. And then we have the transporter that will transport drugs through the transmembrane via active or passive transport. And we have the modifiers that will modify actually the activity of ADME genes based on the interaction with or that will regulate the transcription level of these ADME genes by interacting with particular regions in the DNA. So in total, there are probably 340 genes, ADME genes, that belongs to all these classes. They are spread all over the genome, except maybe for the Y chromosome here. And you may maybe find some discrepancies between the bibliography, because some of the researchers, they consider only 300 genes, but based on this article, we're now at 340 genes. So the main idea here is to be able to characterize these genes in African population. And by characterizing, I mean, of course, that for each position we want to detect the polymorphic sites, both in the coding regions and also the non-coding regions. And also we want to be able to detect the effect of this variability in African population. Now, there are actually 2 main approaches to characterize these pharmacogenes or these ADME genes. Well, actually, let's say there are 2 main tools that will allow us to characterize these genes. So at the genetic level, we can, of course, do the sequencing. Most famously, maybe the next generation sequencing could be an exome sequencing or could be a whole genome sequencing. So this is very easy now, and this is very straightforward. The cost of the whole genome analysis, I think now it's at $1,000. So you would be able to characterize both the encoding and the non-coding regions. But also, so this will allow, of course, to detect the variability in the genome. But then also you want to link the variability with the phenotype. And then you will employ maybe some biochemical methods or some functional characterization. And basically what you are doing is that you measure an activity. Say, for example, that you want to detect a secondary metabolite in the blood that is the product of the metabolization of one drug. And then you want to follow that during at different concentration of the drug or at different levels of time. And then you dress up these association laws, and then you apply a statistical model. And from this statistical model, you will be able to associate the genetic variability and the biochemical effect. So these are maybe the 2 working horses that we are currently using in order to study the pharmacogenomics. Now in Africa, I think, if I'm not mistaken, maybe the pharmacogenetics has gained a pace at the start of the year 2000, actually. And basically, this is a typical study where we characterize SNPs, or we characterize also genetic variability, and we associate the genetic variability to a phenotype. Let's say, for example, a drug efficacy or drug toxicity. But then also we now I think we conducted the first whole genome study, or at larger scale. So in this study, and we have published that in 2020. This is, I think, the only study now that include a large cohort from sub-Saharan Africa, and they try also to characterize the pharmacogenomic landscape in these populations. So, so we spoke about the different positions that we want to characterize, but actually, I think if you are already doing pharmacogenomics, I think you may heard about the notion that, or the concept of star alleles. So what are actually star alleles? So if we imagine, for example, that we have here a gene, and we have here different loci. So at these loci, polymorphic loci, we'll be able to characterize all the genetic variability at these positions. But then these positions, actually, they don't segregate separately or independently. So actually, they segregate together. So because of the linkage disequilibrium. And so we call these position a haplotype. This is a common jargon in genetics, but in particular in pharmacogenomics, we call them star alleles. And because you inherit 2 copies from your parents, then you have 1 copy from the father and 1 copy from the mother. And then you have here what we call a diplotype. So the diplotype actually will determine the fate of or the phenotype of a particular drug that you take, for example, to cure a disease. To cure a disease. And based on this diplotype, actually, you will be able to metabolize, for example, very rapidly a drug, or very slowly, or you may also, unfortunately, you can also have some side effects. So as an example, we have here the drug codeine. It's very common drug. It's an opioid analgesic commonly used in Africa. So codeine is metabolized by cytochrome 2D6, CYP2D6, which exhibits significant genetic variation across Africa. Now, if we try to characterize the different or the diplotypes of the star alleles in Africa, some particular haplotypes or star alleles, they show particular prevalence in Africa. So, for example, here the CYP2D6 star allele 4, it's very frequent in European. It's at least at 18% frequency. But in Africa, it's only characterized at 0.03. So this means that if, for example, you want to, you have a pharmacogenomic clinical guideline that will say, for example, that whenever a patient is taking a codeine, then you must do, for example, a genotype, or you must characterize the CYP2D6 star allele 4 in this patient, because we have 18% of frequency for this particular haplotype. So this will apply in Europe, but maybe not. It's not applied in Africa. But still, we have a low frequency. There are still some people who have this star allele, but then in terms of frequency, or in terms of reliability or efficacy, or in terms also of economy, that may not be the correct choice for Africa, and vice versa, because sometimes also you will have a star allele that is very frequent in Africa, but is very low in Europe. And this star allele should be, or must be, taken into consideration when you address any guidelines, clinical guidelines in pharmacogenomics. So, and basically, this is how we work. So basically, we can adjust the pharmacogenomics guidelines based on a set of evidence about the function and impact of a diplotype and the allele frequency in the population. And this is maybe how we can suggest if certain guidelines will be implemented in the clinic or in precision medicine or not. Okay. So then if we want to do this, if you want to establish clinical guidelines, what type of information we need? So as usual, like we said this before, we need the genetic characterization. We need to detect the general landscape or the general pattern of variability in the population under study. But also we wanted to associate the certain variability to the functional properties. So here you have the genetic part, and here you have the functional part. And both of them, they are essential in order to establish these guidelines. Okay, so now we have the recipe here. And you may say here, okay, that this is an easy affair. But the thing is, it's not, because biology actually is very complicated. And what we show now, for example, an association between, let's say, drug efficacy and a certain haplotype could be also not so accurate or not so obvious, because different other factors may intervene. So we may generate some very basic rules based on some limited observations, but then in the real world, it's very complicated. And then the question here, could we can we scale this to the level of the genome? And what are the different levels of complexities in pharmacogenomics that we need to consider in order to get accurate results or get accurate observations?
Donation, please. Yes,
Sorry?
I don't have a bag to put them.
Okay,
Okay. All right. Okay, so the question now is about the complexity. And this is what I'm going to show in the second part. So what are the different levels of complexities in pharmacogenomics? So the first level of complexity is, of course, the Mendelian association. You have here different types of drugs. These have different also chemical structures or chemical properties. And you may find, for example, a drug that depends, or the metabolization of a drug that depends on certain positions in the genome. You may have also some drugs also, they depend on a single position. So it depends really on the type of drug. This is a common concept. So what basically it says here is that the observed phenotype could be associated to a limited number of positions in the genome, and you'll be able to predict easily the phenotype based only on several positions. So, for example, the adverse effects caused by codeine are associated to many genes such as OPRM1, CYP2D6, SLEC22A1, and others. This is very common. The second level of complexity is the CNV, copy number variants. Basically, you have a drug here that depends on the or which fate depends on the presence of certain genes. And then these genes actually, they could be spread all over the genome, having multiple copies. So you don't have only one copy of a gene here, you have multiple copies. And depending on this number of copies, the phenotype will be determined. So for example, CYP2D6 star allele 1 times n, which means that CYP2D6 will have n number of copies, is associated with respiratory insufficiency when treated with codeine as compared to CYP2D6 star allele 1, star allele 1. So this is here the genotype. So multiple copies for CYP2D6 is bad, but then if you have one copy on each from each parent, then that's fine. So this is the second level of complexity. The third level of complexity is the polygenicity. And the drug here actually depends on the additive effect of different positions in the genomes. And these actually, they will probably be associated with mild to low level of metabolizations or toxicity, for example. And based on this position, you'll be able to generate, for example, a linear model that will predict the phenotype based on the frequencies of these positions here. And for example, here we have most of the drug response phenotypes are extremely polygenic, and in some drugs, moderate and low effect variants are contributing to the most to the pharmacogenomics trees with high heritability. And I really suggest to maybe to look to this bibliography in order to understand better this phenomenon. Now, if we talk about polygenicity, of course, we're going to talk about heritability. And so this is the fourth level of complexity. So heritability is determined by your genome, but also you have other factors here. And these factors actually could be determined by multiple covariates. So for example, a person is not isolated from its environment. So he's living, he's having a way of life, he's eating also specific food. So he's really imprinted in his environment. And so if we determine the different levels of, or the different factors, or the different covariates, and associate that to the genome, then we may also determine the fate of pharmacogenomics phenotypes based on that. And as example, for example, you have the exposure to pesticide may affect drug metabolization under the effect of phase 1 and phase 2 enzyme, which means that it's not enough to know only the genotype or the star allele of certain enzyme. You need also to have a good overlook on the exposome in order to predict well your phenotype. Okay, so you may see now from this picture that there is a lot of things to cover, and only the I think here at least only having a glance to the genome is not going to be enough in order to predict the effect or the phenotypes in pharmacogenomics. So that leaves us with two choices actually, because you have to make some sacrifice here. So we have two choices. The first choice is we can go big, but only be able to generate descriptive knowledge. And I'm going to show later what I mean by this. And the second part is we can go small and precise, but we won't be able to scale to the African genetic diversity. Which means that you will be able only to do properly certain association studies for limited number of loci or limited number of genetic variability in order to characterize well your the phenotype, and also in order to be able to implement maybe some certain clinical guidelines. Okay. So this is about the complexity. Now we are going to talk about what we did for this part. This part here. Oops. And what I meant actually by this part. And I'm going to go next to stating the problem from this talk. So like I said before, the first and I think also the only genome-wide study of ADME genes in Africa was performed by George Darosha et al. And we have done that from different sources of data. So basically what we did is that we collected several data sets from data gatekeepers or collaborators. And this covers a lot of African countries. But we have here an imbalance between maybe the South and the East. We have also some decent number of samples from West Africa. As you can see here, from Nigeria, we have 53. We have 33 from Burkina Faso. But still also we have some low coverage from Nilo-Saharan countries and Eastern Africa. And as for this sample from Algeria, it was discarded because it's an outlier. But in total, we have 458 samples. These are whole genome sequencing data with high coverage. And to that, we added the 1,000 genome data set. And so we aggregate all together and we did our analysis. So for this particular 458 samples, we first did a PCA. And from this PCA, actually, we did also some clustering analysis as a first step in order to determine what are the groups that could be emerging from this study. And from that, we have identified 4 main groups. So the first group is the far western group. The second one is the Western group. The third one is the South Center group. And the largest group is actually the Southern African group. And you can see here there are some outliers, and this is maybe due to the Khoi and San component in our data set. But basically, we have subdivided this in 4 population groups based on all the positions in the genome. And as a first step, we had all the genetic variability, all the positions for these ADME genes, and we identified what we call high impact variants. So these high impact variants were identified based on computational criteria. And so when I say a high impact variant, means that these variants are supposed to show a significant impact on the function of the protein. And if you look to this Venn diagram now, you may see that the largest number of these variants are actually unique to each population group. So I have here 133 in Southern Africans, 109 in Southern Central, and so on, so on. Okay, so these are really unique. And also, if we look to the frequency, we may find also that these unique variants most of them are actually rare, because they show a low allele frequency for all the population groups. So in conclusion, most of these high impact variants identified using computational tools are rare and unique to each population group. So what this may have as an impact. So now if we, for example, imagining that instead of having like 458 individuals, we increase the level of sequencing. So say, for example, that we start at this level at only few samples here, and then we increase each time the number of samples. Each time you do that, actually, you will be able to identify some rare variant, and you can also identify some unique variants in these population groups. Okay, so each time you will actually identify some other population groups and, of course, some unique rare variants. And each time also you increase the level of sequencing in the population in Africa, you will be also able to detect maybe unique actionable variant or unique rare putative actionable variant. So really what this is tells us is that the level of details in terms of genomics of ADME genes in Africa is still at low coverage now. So wherever if we decide, for example, to have more sequencing, then we will be able to identify other types of variants. And the variants that were identified in our study are not actually enough in order to say that we have a representative set of variants from which we can do some or can establish some, for example, clinical guidelines. This is the first thing to notice. The second thing to notice, and what we did also in our study, is to actually think about the population groups as proxy population to each region. So, for example, if I'm talking about Southern African, so Southern African could be, maybe they could be representative of Mozambique, Botswana, and maybe also Zimbabwe. I don't know. Okay, so that's what we think. Maybe this population or the population groups could be representative for all the regions here. And what we found is that's not true, actually, because if we see here, for example, the genetic distance between Southern Africans and Far Western, it's actually the Far Western and the South Central is 0.0043. This is the FST measures. And it's almost the same between the Far Western and the Western, although the Far Western and the Western are very close in terms of geography. So really this notion of proxy population maybe does not exist in Africa, particularly for the ADME genes. So that actually has an impact. So if we think, for example, that we have here a population, and for these subpopulations, you have for each one a particular drug that is applied and could be actionable actually. So whenever you increase the level of granularity, which means the level of granularity is actually identified new variants in Africa. So whenever you increase that, you'll be able also to increase the number of the rare unique variants that are not representative for the proxy populations. And so you end up with population groups that have actionable variants, and some other population groups or unique individuals that don't have really actionable variants. And this is really causing, I think it will cause some ethical challenges and also legal challenges and economical challenges, because you won't be able really to deliver an efficient and a robust therapeutic guidelines for these individuals for which you don't have the time to characterize, or you won't be able to characterize because of the effect of granularity in your population, and also because you don't have any proxy population based on which you can dress up your guidelines. And this is why actually we have a very now big challenge in Africa. So through all during the last years, actually, what we've been doing is concentrate on this part, on the discovery part here. So we discover variants, we characterize some of them, and some of them will cross this actionability threshold. And this is the actionability domain. So all the variants here, all the information here are actually translated into clear clinical guidelines. And what we're doing now is just cluttering and piling up data at this domain, while we cannot really, we are not able to go to the other domain because of the different challenges that we have and that I have explained in terms of complexity and in terms of the structure of the African population, but also for some other reasons that I will explain later. So basically, the problem is could be stated as follows. So our ability to identify interesting variants surpasses our ability to identify and put into action real interesting variants. And with this, I conclude the third part. And now, I know some of you here are actually sacrificing their Saturday time for bioinformatics and artificial intelligence, which I'm hoping in this part I'm going to explain. So I'm not going to explain, of course, bioinformatics and artificial intelligence, but what these both subtopics could do in order to tackle this problem. And I think if we have a very cartoonish or a very drafty framework, we can say that in order to be able to have clinical implementations, it is really the holy grail of everything we are doing in pharmacogenomics and in precision medicine. Then we have first to get a robust system of computational prioritization. So this system will take the data from high throughput genetic characterization. And here I'm not talking even about other types of analysis like functional analysis. So only high throughput genetic characterization. And this system here will be able to filter out the most important information. And this information will then go through a robust system of functional prioritization. And from that, we can also lower down the complexity of a problem until we can actually, the relationship of genotype and phenotype could be established clearly in order to be implemented in the clinic. And here comes actually the contribution of bioinformatics and artificial intelligence, because both these tools could be very important in order to prioritize variants and filter out what is only important to attain this holy objective. So about artificial, let's start first about artificial intelligence. What artificial intelligence could do for you? So it could be used to discover new patterns in high throughput data. So there are some hidden patterns in each data set, and you need really to apply some robust algorithm in order to discover or identify these patterns, because they could be associated to some very interesting observations in the real world. The second thing, you can also apply artificial intelligence to mine the biomedical literature using natural language processing, because now we have millions and millions of abstracts and papers. They appear each year, and in some of these papers, there are also some hidden information that you need to discover and identify throughout this technique. The third thing that you can do with artificial intelligence is you can apply statistical modeling for aggregated risk predictions. Remember the complexity of the different or the multiple covariates. So we can, through or using artificial intelligence, aggregate different types of data and then try to apply some statistical model in order to understand the relationship between the covariates and the genome and also the phenotype. And fourth, you can do the monitoring of healthcare data in real time. And this is very important because what implies is that you will be sitting on your desk, basically, and then you have a system that will capture the essence of what you're trying to or what you want to capture. So, for example, you want to capture events of toxicity in patients, or you want to capture, for example, some very interesting phenotypes that needs more focus in research, or also a more focus on clinical monitoring. So you'll be able to do all this with artificial intelligence. And I'm sure this is not the only thing that could be used. There are some other instances and other cases or showcases of artificial intelligence in pharmacogenomics. And I'm very interested to hear from you maybe later about that. So if we talk about now the artificial intelligence in a very generic way, or most of the people, I think they heard about supervised learning and the unsupervised learning. So sorry, here I have made an error. So this should be a supervised learning. It's correct. This should be unsupervised learning, not supervised learning. So in both of these approaches, actually, you need for example, for supervised learning, you'll need a matrix of features, okay? And you need also to label your data. So for the matrix of features, you will subdivide your data to a training data set and the testing data set. And you have here the labels of the data telling you, for example, that for this patient, you have, for example, a toxicity, or for this patient, you have maybe, I don't know, maybe low level of second metabolites, or anything, could be anything, okay? Or could be just a discrete label, could be a dog, cat, anything. But the basic idea here is that you need a matrix of features and you need data labels. And you will feed that to a statistical model, and this statistical model will be able to predict, based on this matrix of features, the classification of each one of your data. And you can apply your statistical model after the training on real case data to do some interesting predictions. So this is the supervised learning. For the unsupervised learning, you don't actually need the data labels. You need only the matrix of features. And again, you will apply a statistical model, but in order to detect the patterns in the data. So this is not a course about machine learning, but just to tell you here that the most important part, and if you know, of course, the principle of garbage in, garbage out, the most important part here is the data. So where can we provide this data? And this is actually a very big challenge in pharmacogenomics, and not only in pharmacogenomics, but I think It's in omics studies in general. So let's remember, for example, that we have our main data are sequencing data. So if you have sequencing data actually only sequencing data, you won't be able to do a lot of things with that. Only some exploratory analysis. But if your data lacks different types of informations, like age, like phenotype, like different other metadata, or if you don't, if you didn't carry the annotation in a very robust manner, or if you are missing functional data, if you didn't have any plan of sustainability for your data, and if your data is not fair, which means findability, accessibility, interoperability, and reusability, then your data will be killed, and it won't be very useful to do, for example, artificial intelligence and machine learning, or be applied for secondary types of analysis. And this really now should be addressed not only in Africa, but I think for all the researchers, because what we were taught as academicians or as a scientist is that we are the engines of knowledge. So we generate knowledge. That's our job. But then there is a second job, and I think it's very important, is to be able to create the data for and provide that to the community. So if we will be able to do that, which means not only the sequencing data but also the metadata, then our effort will be very valuable in order to be used for artificial intelligence applications. And I think the best example for that is the African Genomic Medicine Portal. This is an initiative that was nascent as part of the activities of the H3 Africa BioNet. And basically what we did is we create all the biomedical literature to find associations, disease association, and also pharmacogene phenotype association in Africa for different African populations. And then from this, we established this platform. So if you are interested in using that, you have here the link, and you have also the paper that described the different steps that we took in order to develop this tool. And this is really the effort of a large community of creators that englobes the bioinformaticians, geneticists, medical doctors, and many other contributors. Now, I'm going to just show you some examples here of the applications of artificial intelligence and pharmacogenomics. So this is one of the cases that we used in order to discover some of the patterns or the pharmacogenes that are most relevant in TB and malaria drugs. So we used actually what we call a PGX corpus. So basically, this is a series of texts that we constructed in order to train a model. And this PGX corpus was then used to train the model and to classify the abstracts and the different phrases in the abstract in order to detect the drug-gene association. And for that, we used what we call a tagging system. So a tagging system takes a phrase or a sentence, and this tagging system will be able to detect the biological entities, or for example, azoniazid is metabolized by polymorphic N-acetyltransferase NAT2 enzyme. So the tagger actually is able to detect that this is a drug, and also it was able to detect that this is gene. So not only this one, all this here is a gene. And then from this classification, we can detect, or we can detect the association between the drugs and the gene. And by establishing a predictive model, it was a very accurate predictive model. We were able to identify 24 common pharmacogenes between malaria and the TB. starting from more than 72,000 and more than 200,000 abstracts in bibliography. Not only that, because we also were able to identify 9 less known genes that are very relevant to TB and malaria pharmacogenomics in Africa. The second use maybe of AI that we did, we actually
sorry.
Yeah.
Sorry, Doc. Please, you may consider rounding up so that we can have a few minutes for some questions. Thanks.
Yeah, sure. Okay, so here we have an example where we use both the information of the chemical structure of the drugs and the sequence of cytochrome P450s, and we use deep learning model to classify the associations between the drugs and their targets. I'm not only going to take, not going to talk about this. Okay, then I'm very excited actually that we have also some very good progresses in neural network, because before for deep learning, we don't have the ability, or we have very few ability to explain the models, the predictive models. So now with the coming of Kolmogorov-Arnold networks, we are actually able to add some explainability to deep learning network, deep learning method, and thanks to this, we will maybe be able to take into consideration the complexities, different levels of complexities in pharmacogenomics. Now, in terms of the contribution of bioinformatics, actually, just some important things to notice. And one of the major area of research now is to tailor some specific scoring functions or scores or predictive score for the variant effect prediction in pharmacogenomics. And we have been able to calibrate some of the pipelines based on some of the research that was published before. Known also that some of African researchers, they did their own solutions, or they did develop their own solution to do that. One of these tools is Stellar PGX, developed by David Twasigame, and this tool is also available on GitHub, and you can definitely use it and contact David to use it. This is really tailored for pharmacogenomics analysis. And in terms of the usage of multi-scale approaches, we were able to integrate different types of analysis. The main one is the single point energy mutation. So basically, we take the structure of the proteins of the ADME genes, and then we characterize the genetic variability based on biophysical measurements. And we were actually very successful to do that based on a model, in a linear model that don't cost a lot in terms of computational power. And this is the work that you can verify also by Nklamulo, Koza et al. We used also some other techniques like the molecular dynamics. This is an ensemble-based method where you generate different conformations for a protein, and then we were able actually to characterize the genetic effect or the impact of the variants based on purely on energetic scores. And also we were being able to detect some very interesting properties like the binding of ritonavir with artemether and so on, so on. You have also to be very excited with what the future will bring, because now we can, and with the coming of the new age methods like the artificial network and the LLM models, for example, we can generate conformation ensembles not with using molecular dynamics, because molecular dynamics requires a lot of computational power. This one maybe is not, will not require too much computational power. We can also now predict the effect of or the interaction of the drug with its target simultaneously during the folding event, which is very good because we won't only be able to do that in a rigid manner. And also because we have AlphaFold 2 and now we have AlphaFold 3, we can do or perform genome-wide annotation-based physical models. And we'll be able to do some assessment of this method, but only for some of the genes. So the conclusion now. And all right, so okay, so for the conclusion, like I said, we need to go from the discovery domain to the actionability domain. And I don't think that the strategy of first sequence and then ask question is going to work here. Rather, we will have to do some intelligent sequencing. So the by intelligent sequence means that we do have this, we will do the sequencing, and also we will do the phenotyping or intelligent phenotyping or deep phenotyping. So we're not only gathering the sequencing informations, but also the metadata. The second thing is that we will have to get our H3 Africa like for functional genomics. And this means that we need to advertise and also to advocate the importance of functional studies in Africa, and maybe we need to have some very strong consortium that will promote techniques such as deep mutational scanning or gene editing or protein expression or high throughput calorimetry assays. And this is very important in order to characterize functionally the African variants. Also, we will have to be aware of our part as a curator, not only knowledge generator. And by that, you need to maybe to join some African networks in order to participate in these efforts. And then also we need to promote the different studies of that will try to establish interesting or important statistical model in order to be able to tackle the complexities of the data. And both of these actually go in parallel. So these guys actually, they will be able to feed data to these guys in order to characterize well the genetic variability of in Africa. And with that, I am able to reply to your questions.
Thank you. Thank you so much, Othman, for this great talk. So I think we'll have some time for some questions, brief. So let me see if I have a hand or a question in the Q&A. So now I do not have any question there, but from the great insights that you've actually showed on this particular topic, doing justice to it, looking at the complexities that you've outlined as going forward, we have to overcome to be able to actually bridge the existing gap. What I'm interested in particularly is the clear difference between the discovery domain and the actionability domain. So where are we now when it comes to Africa? In terms of, because the goal is that we'll generate this knowledge towards precision medicine, have data to actually contribute to clinical decision support systems in our health sector. How far are we from translating any pharmacogenomics data that we have now to the clinical settings? How far are we?
I think actually in Africa we are in a good position. And now, if you talk about this graph, we are still here. But in general, we are in a very good position. Except that I think many teams, they are working in silo, and what we need now is a coordination of the efforts in order to valorize each team's or each country's effort and go to the actionability domain. The other issue is that here different countries, they have different challenges, so we need maybe to like I said, we need to discuss this as a community And we need to prepare another persona of scientists that will not only generate data, but also will generate, not only generate data for their teams, but only will generate data for perspective usage and perspective plans for their team and also for the community.
That's very interesting. Then the next one has to do with when it comes to actually overcoming these complexities, then the way forward is the artificial intelligence. So now how do we kind of demystify the complex models so that it becomes transparent for the generation of this clinical framework and guidelines, and also for the clinicians, or those at the forefront to understand us, because that's kind of like a black box. That transparency is a myth that is hampering the translation of let's say models and other things to the clinical setting. So how do we break it down to make our health workers understand in terms of training? Any ideas so far on that? Thank you.
So in terms of artificial intelligence, the experience now shows that many of the established predictive model, they are not easily implemented in clinics. So you'll find maybe 80% of the applications are for image processing, but for genomics, it's only few of them actually were approved by the FDA, for example. And the problem is that maybe this type of applications, they're not exclusive to the IT. So we need maybe a more contribution from the clinicians, more contribution from scientists. And I had to say this because I think there is a trend now to go after the buzzword like artificial intelligence and precision medicine. And everybody now is trying to data science everything, which is not a very good thing, because what I believe now is we should give more power and more weight to the experimental biologists, because this is what we are going to, and because through this we are going to construct a core data, and based on this core data we may later move to the establishment of very accurate and very reliable artificial or predictive model. And the other thing about the predictive model, I think like I said, the recent progress in artificial intelligence and the Koronkov-Arnold approach, it will add more explainability to the data. So we will be able in the future maybe to tell from thousands of features or thousands of parameters what are the different parameters that will account for a certain phenomenon or for certain association between the drug and the genotype and the environment. So the future is really bearing very exciting things, and we should be prepared both as bioinformaticians or biologists to the futures. And really the key thing here is to really change the mindset.
Okay, that's great. So we have a question from the chat box, and it has to do with, I think, so I'll read it as it is. Which teams, so with it comes from the background that research groups are reluctant to share data. When researchers generate data, they are reluctant to share the data. And so where do you see like openness? For instance, you are a key promoter of open science and open research. Openness in research environment as playing a key role in reaching this goal, so that we can benefit from this precision medicine and the AI and what's the, considering the fact that when we come to Africa, our priorities are clearly different. The burden of diseases are high, and so if we continue to hold on into data as silos, individual, what will be your take on that? Thanks.
So I think the most important aspect to understand.
Where do you see the the problem of people holding on to data, being reluctant to share data between research groups contributes to us moving from this discovery to actionability domain.
Yeah. So the problem of data sharing actually, I really don't want to judge people, because those who prefer not to share the data, they have their own reason. But then the most important thing is to clarify that sharing the data actually, it's not a grandstanding, moral grandstanding. It's not to say that I'm better than you and I'm a very moral person, or no. It's actually, when you share data, you create the opportunity for yourself and your team. And the second important thing that you need to also to consider is that it's okay to protect. It's actually very important to protect the confidentiality of your participants or your patients, but also you have the duty or each one of your patients have also the right to access to science. So, and they have also the right to be delivered, or to have the most efficient and the most reliable treatment in the world. And if you are going to keep the data only for yourself, then I don't think that will profit the patient the most. Good thing is that we have some legal and ethical frameworks that are well established, even in African countries, and we need only to follow these frameworks. Some of them actually they're not perfect, but they could be used to conduct this type of research and protect the data. So really, the key part here is maybe we need to see the interest of sharing data, not from a moral point of view, but from interest point of view. It will benefit your community, it will benefit your career, it will benefit your team, and also it will have a very significant impact to your research in general.
Okay, thanks. So I think Rim, do you have something to contribute? Because we've already run out of time and I wanted to run it up. Was your hand up? Okay, then that's fine. So Dr. Othman, we are very privileged and we are grateful for your time this afternoon and sharing this insight on a key and timely topic like this. So clearly, from what you've shared with us, we can actually appreciate the tremendous potential for advancing in personalized medicine when it comes to targeting pharmacogenomics. Not only on improving the patient's outcome, we've also highlighted some challenges like even data quality, and also you've mentioned ethical considerations, regulation also, then even the computational demands, which is another topic, maybe another day to discuss the demand for the computational demand when it comes to this. And before we can now move on to this clinical adoption for our health benefits. So we are very grateful and we thank you for your time.
Thank you.
And we thank everybody for also making time for this webinar. And this is a webinar series that has just started for the year. So it's going to be monthly and it will run every month. And kindly follow our social media handles and also the website to also get the dates for this upcoming webinar. So we thank everybody for making time, although it's a Saturday. We are grateful. I think I have a comment. Let me just be sure before I run it up. Okay. So thank you very much for making time for us. We are very grateful for this insight. We will contact you later for any further discussion. Thank you very much. And,
thank you. Please contact me if you need to develop anything in this presentation, or if you think also you want to share any ideas with me also.
Thank you very much. That is great. So then, bye-bye to everyone. See you again on our next webinar. Bye-bye.